is a disclaimer for the viewers. I once again welcome you all in the session of novel drug delivery system. Up till today, we have finished more than 7 to 8 chapters from the novel drug delivery systems. In the today's session, we are going to discuss the new topic, that is a targeted drug delivery system. Content of the topic, that is a targeted drug delivery system, we are going to discuss the concepts and approaches, then advantages and disadvantages, introduction to liposomes, niosomes, nanoparticles, monoclonal antibodies, and their applications. Learning objectives. So you will be able to define targeted drug delivery system. What is mean by the targeted drug delivery system? You will be able to write the advantages and disadvantages of targeted drug delivery system. Then you will be able to describe concepts of targeted drug delivery system. You will be able to formulate targeted drug delivery system using different approaches. Then you will be able to describe concepts of liposomes and niosomes. You will be able to explain concepts of nanoparticles and monoclonal antibodies. And you will be able to discuss various applications of targeted drug delivery system. I have set the 7 learning objectives which will cover the all content of the targeted drug delivery system. In brief, we are going to discuss the definition of targeted drug delivery system, its introduction, need, then advantages and disadvantages of targeted drug delivery system. We are going to discuss certain concepts related with the targeted drug delivery system, then different approaches for the targeted drug delivery system. Then furthermore, we are going to explore the concepts related with the liposomes, niosomes, nanoparticles, and the monoclonal antibodies. More specifically, we are going to discuss the advantages, disadvantages, methods, evaluation and applications of the liposomes, niosomes, nanoparticles, and the monoclonal antibodies. So these are the learning objectives which I have set for the targeted drug delivery system. Let us move towards the introduction to targeted drug delivery system. Before starting the introduction to targeted drug delivery system, I would like to mention here, I have made approximately 5 parts related with the targeted drug delivery system. Very first part, we are going to discuss only the concepts related with the targeted drug delivery system. Second part, that is related with the liposome. Third part, that is related with the niosome. Fourth part, that is a nanoparticle. And fifth part, that is a monoclonal antibodies. So it becomes very easy for you to understand the things. So let us move towards the introduction to the targeted drug delivery system. As a targeted drug delivery system, it is said to be the smart drug delivery. As all of you are aware about the disadvantages of conventional drug delivery system, as there is a large fluctuation, large dose is required, isn't it? And when we are formulating or we are targeting the drug to the specific site, it is possible to minimize the dose size, frequency of administration, and the toxicity of drug. So sometimes it is called as the smart drug delivery. It is a method of delivering medication to a patient in a manner that increases the concentration of medication in some part of body relative to others. For example, say if we want to target the colon, it is possible to prepare colon targeted drug delivery system by using the certain polymers which are degraded into the colon. More precisely, already we have discussed the gastroretentive drug delivery system. If we want to target the stomach for the specific infection, it is possible to target the specific site, that is the stomach, by using the different approaches or by using the different polymers. So it is a method of delivering medication to a patient in a manner that increases the concentration of the medication in some parts of the body relative to other. It plans to overcome some disadvantages of the conventional drug delivery system. All of you are aware about the disadvantages of conventional drug delivery system. So it is possible to overcome certain disadvantages of the conventional drug delivery system. This system would be loaded with a drug and targeted to specific part of the body where there is slowly diseased tissue, thereby avoiding the interaction with the healthy tissue. For example, if if we want to target the cancerous cell, it is possible to target the cancerous cells or the infected cells by using the concept of the targeted drug delivery system, or by using the different approaches of the targeted drug delivery system. So automatically, whatever the local side effects are systemic side effects are there, it is going to be minimized in the case of targeted drug delivery system. The goal of targeted drug delivery system is to prolong, localize, target, and have a protected drug interaction with the diseased tissue. The main goal of the targeted drug delivery system is to prolong, localize, target, and have a protected drug interaction with the diseased tissue only. It implies for selective and effective localization of pharmacologically active moiety at pre-selected targets in therapeutic concentration while restricting its access to non-target normal cellular lining, and thus minimizing the toxic effects and maximizing the therapeutic index of the drug. So the targeted drug delivery system, it is a selective, it is a there is a effective localization of pharmacological active moiety at a pre-selected target in a therapeutic concentration, while it is going to restrict its access to non-target normal cells, and thus minimizing the toxic effects of the drug and maximizing the therapeutic index. So this is about the targeted drug delivery system. Let us move towards the need of targeted drug delivery system. And here we are going to discuss the need in terms of the pharmaceutical reasons, in terms of the pharmacokinetic reasons, and in terms of the pharmacodynamic reasons. In the case of pharmaceutical reasons, there is as you know that there are so many drugs are there which are having a low solubility, and there is an issue of the stability. So these two problems can be minimized with the help of the targeted drug delivery system, that is related with the drug instability and the low solubility. Then the pharmacokinetic reasons, there are few drugs are there which are having a short half life. Then there are certain drugs are there which are having a large volume of distribution, and there are certain drugs are there which are having a poor absorption. So whatever these issues are related with the pharmacokinetics of the drug can be minimized with the help of the designing and developing the targeted drug delivery system. Then pharmacodynamic reasons, that is a low specificity towards the tissues or cell, or the low therapeutic index. So these are the certain things or reasons uh, which gives us the idea why to design and develop the targeted drug delivery system. That is the pharmaceutical reasons, pharmacokinetic reasons, and the pharmacodynamic reasons. Let us move towards the advantages of targeted drug delivery system. Now you are aware about What is mean by the targeted drug delivery system? We are targeting the specific organ. It is possible to target the specific tissue. It is possible to target the specific cell in order to increase the efficacy of the drug. Let us move towards the advantages of the targeted drug delivery system. It offers the so many advantages. Very first one, it is going to reduce the dose of the drug. It is going to reduce the dosing frequency. It is going to reduce the drug side effects and the toxicity, and also it is going to minimize the fluctuations in the plasma drug concentration. So there is a reduction in the dose of the drug, reduction in the dosing frequency, reduction in the drug side effects and toxicity, and reduction in the fluctuation in the plasma drug concentration. Apart from these four advantages, there are so many other advantages are also there. It is going to avoid the hepatic first pass metabolism of drug. Thereby, it is going to improve the efficacy of the product. We are getting the more uniform effect of the drug, as here specifically we are targeting the organ, cells, or the tissue. As selective targeting to infectious cells than the normal cells. So it is possible to protect the normal cells. It is going to maximize the therapeutic index of the product. There is a positive effect on the permeability of the proteins and the peptides. So it is possible to design and develop the targeted drug delivery of the certain proteins and the peptides. And finally, it is going to improve the drug bioavailability. So these are the certain advantages of the targeted drug delivery system. Let us move towards the disadvantages of the targeted drug delivery system. Like advantages, it is having a certain disadvantages also. As very first one, that is high cost. It's not that much easy to design and develop the targeted drug delivery system. So it is having a high cost, which makes productivity more difficult. There is a reduced ability to adjust the dosage, as here we are developing certain novel carriers to target the tissues. So there is a reduced ability to adjust the dosages like a conventional drug delivery system. There is a rapid clearance of the targeted system. Then there might be chances of immunoreactions against the carrier system. Here we are using the certain carriers, and there might be chances of the immunoreactions against these carriers, and these carriers gets easily cleared from the body. Insufficient localization of targeted system in the tumor cell. More specifically, the targeted drug delivery systems are designed to treat the cancer, but there might be chances of insufficient localization due to certain reasons. That is one of the disadvantage of the targeted drug delivery system. Also, furthermore, there is a diffusion and redistribution of released drug. If the released drug, it is going to be redistributed into the systemic circulation, it is not going to be targeted well. It requires highly sophisticated technology for the formulation. If we want to design or develop certain carrier systems which requires the highly sophisticated technology, it requires skills for manufacturing, then storage, and the administration. It's very difficult to maintain the stability of the dosage form. For example, liposomes. Then there is a low drug loading in the formulation, as here we are using the certain carriers. So there might be the low drug loading is the one of the major issue related with the targeted drug delivery system. And already we have discussed that is a difficult to target the tumor cells. So these are the certain disadvantages of the targeted drug delivery system. So before designing and developing of the targeted drug delivery system, one need to consider all these drawbacks or lacunas of the targeted drug delivery system. So let us move towards the properties of ideal targeted drug delivery system, how it should be actually. So targeted drug delivery system, it should be non-toxic, biocompatible and biodegradable. As here we are using the novel carriers to formulate the targeted drug delivery system, and we are utilizing the so many excipients. So there might be chances of the toxicity of these excipients, and these excipients may not be compatible or it may not be biodegradable. So whatever designed targeted drug delivery system is there, it should be non-toxic, it should be biocompatible and the biodegradable. Then it should be non-immunogenic, it should not produce any immunogenic response in the body. It should be physicochemically stable in vivo as well as in vitro. So whatever the formulation is there, it should be stable in vitro situation as well as it should be stable in the vivo situation also. It should have the controlled and predictable drug release. So whatever the drug release is there from the targeted drug delivery system, it should be controlled and it should be predictable. Drug release should not affect the drug action, it's the pharmacological action. It should have minimal drug leakage during the transit. Suppose that we have given the formulation by oral route of administration, and as you know that through the systemic circulation it reaches to the target site, or say for example we have given it by the parenteral route, so it has to travel the veins and arteries. So during this traveling period, there should be the minimal drug leakage. The preparation of targeted drug delivery system, it should be easy, reproductive, and the cost effective. It should have restricted drug distribution to target cell or tissues or organ, and should have uniform capillary distribution. So whatever designed and developed targeted drug delivery system, it should have the restricted drug distribution into the target cell only. Whatever the carriers which are used should be biodegradable, or it should be readily eliminated from the body without any issues or without any problem. It should protect the drug from environmental factors such as enzymatic degradation till they reach to the target. So these are the ideal properties of the targeted drug delivery system. Let us move towards the concepts of the targeted drug delivery system and the components of the targeted drug delivery system. So the targeting of drugs to the spatial cells and tissues of the body without becoming a part of systemic circulation, it is the novel idea. If we want to target the liver, if we want to target the spleen, if we want to target the pancreatic cell, so whatever, if we want to target the brain, so whatever the concept is there, it is a something new. The drug can be administered in the form such that it reaches the receptor site in sufficient concentration without disturbing and extraneous tissue cells. So whatever the formulation which is designed, it should be, it is reached to the specific site without disturbing and extraneous tissue cells. So let us see first the functions of targeted drug delivery system. It is having a various functions in terms of facilitate, release, protect, and avoid. So very first one, that is facilitate. It facilitates the therapeutic substance to reach the site of action from the site of administration, where the target site can be organ, tissues, cells, even specific cells or the organelles. For example, colon targeting. For the colon targeted drug delivery system, generally the formulation is given by oral route and there might be chances of release of a drug into the stomach or small intestine. So whatever the targeted drug delivery system which is designed, it should facilitate the therapeutic substance to reach the site of action, that is the colon, from the site of administration where the target site can be organ, it may be tissue, it may be cell or even specific cell or the organelle. It should release the therapeutic payload in its active form in or around the target site, presenting the effective therapeutic levels at the site of action. So whatever the things are there, that is the drug is there, it should be released in its active form at the target site. It should protect the drug or gene from the detrimental effects of environmental factors such as pH or enzymes. So whatever the design targeted drug delivery system is that it should protect the drug from the environmental factors such as the pH or the enzymes. It should avoid the toxicity or adverse reactions of the drug or gene on non-specific normal cells and facilitating administration of lower doses to achieve therapeutic or diagnostic benefits. So whatever the targeted drug delivery system is there, it should avoid the drug toxicity or its adverse reactions to the non-specific normal cells. So these are the certain functions of the targeted drug delivery system. Let us move towards the components of targeted drug delivery system. Before that, let us see the how it is going to be prepared. So targeted drug delivery products are prepared by considering the three important things. Very first one, that is specific properties of the target cell, nature of markers or transport carriers or vehicles which convey the drug to specific receptors, and the ligands and physically modulated compounds. The components of targeted drug delivery system includes the target cell, carriers, and the ligand. So let us see the details of the target cell first. So the target cell is a group of cell in a minority identified to be in the need of treatment. For example, infected tissue. For example, cancerous cell. So target cell is a group of cell in a minority identified to be in the need of treatment, where the treatment it is required. The surface of target cell presents two distinctive cellular elements are considered in the designing of carriers for the targeting via very first one, that is a cell surface antigen, and second, that is the cell surface and receptors. So in the case of first, that is a cell surface antigen exploited in generating cell surface and non-selective antibodies. And in the case of cell surface and receptor, which recognize and internalize the macromolecular ligands associated carriers. So this is about the target cell. So target cell is a group of cell in a minority identified to be in the need of treatment. For example, infected tissue or the cancerous cells. So let us see the types of the target. Approximately, there are 5 types of targets are identified. Very first one, that is the cells. That is in vitro for genome grafting or manipulation of DNA. Second, there is an accessible anatomical compartment. For example, peritoneal cavity. For example, cerebral vesicles, pleural cavity, lungs, and lymphatics. Then macrophages and other phagocytic cells, including Kupffer cells, tissue macrophages, and blood macrophages. Then non-phagocytic cells and the RES, including liver and endothelial cells. And last one, that is lymphocytes and antigens present in the cells. So these are the types of targets, that is cells, certain anatomical compartments, then macrophages and phagocytic cells, then non-phagocytic cells and RES including liver, and lymphocytes and the antigens which are present in the cell. Let us see the carriers. Carriers, it is required for effective transportation of the loaded drug. So its main role is to carry the drug to the target tissue or target site. They are the vectors which sequester, retain drug and transport or deliver it into the vicinity of the target cell. So what is the carrier? It is going to carry the drug molecule from one site to the another site, or from route of administration to the target tissue. There are three types of carriers are there. Very first one is the physical markers, second one is the physiological markers, and third one is the chemical markers. So very first one, that is the physical markers, which are like liposomes and the macrospheres. Physiological carriers, for example, monoclonal antibodies and the erythrocytes, and chemical, that is the prodrug. Let us see the characteristics of the carriers, which are very important because they are going to decide the efficacy of the targeted drug delivery system. So whatever the carriers is there, it must be able to cross anatomical barrier. That is very first important characteristic of the carriers. The target cell must recognize the carrier specifically and selectively. So whatever the carrier which is used for targeted drug delivery system, it should be recognized by the target cell. The linkage of drug and directing unit should be stable in the plasma, intestinal and other biological fluids. So whatever the linkage is there between the drug and directing unit, it should be stable in the physiological environment. After recognition and internalization, the carrier system should release the drug inside the target organ, tissues or cells. And carrier should be non-toxic, non-immunogenic and biodegradable. So this is about the carriers. Let us see the ligands. So what are the ligands? Ligands confer the recognition and specificity upon carrier or vector and lead them to approach the respective target and deliver the drug. For example, antibodies, polypeptides, and the endogenous hormones. So the targeting can be achieved by either direct targeting or external stimuli or chemical modification or by use of the nanocarriers. So direct targeting to the site of action, for example, the topical application for skin disease. It is possible to target the skin directly by preparing the topical formulation. The use of external stimuli, for example, ultrasound, which increases the permeability of the drug. Then the chemical modification of drug to make it physicochemical properties ideal for delivering, which includes the prodrug approach of attaching a pro moiety to the drug. So chemical modification is yet another alternative which is available with us to target the specific cells, tissues and organs. And use of nanocarriers like liposomes, polymeric micelles, polymeric nanoparticles, solid lipid nanoparticles, which can be functionalized further with attachment of the targeting ligand and the antibodies. So these are the ways by which it is possible to target the tissues, cells or the organ or the specific site.